Now the time is 11 o'clock, and I would like to welcome InDex Pharmaceuticals. They released their Q3 and first 9 months report today. And from the company, I welcome the CEO, Peter Zerhuni, and the company's CMO, Thomas Knittel. And I would also like to welcome the audience. And if the audience have any questions, please post them down at your right in the chat, and I will forward the questions either to Thomas or to Peter. So welcome, Peter, and please, the word is yours.
Thank you very much. My name is Peter Zerhuni. I'm the CEO of InDex Pharmaceuticals, and today I'm joined by our Chief Medical Officer, Thomas Knittel. And the topics today is we look at the Q3 report highlights, and then Thomas will provide a deep dive in our previous clinical results. So first, we have our standard disclaimer regarding forward-looking statements, but I thought I would provide a little bit of background on the actual company, InDex Pharmaceuticals. We are a Swedish biotech company. We're developing a drug to be able to help patients suffering from ulcerative colitis to live a normal life. The lead asset is called Copytolimod, and we're now starting phase 3 with that compound. Apart from Copytolimod, we have a broad portfolio of chemically similar assets from our so-called DIMS platform that holds promise in a broad spectrum of immunological diseases, but they're much earlier stage than Copytolimod, so most of the focus is on Copytolimod. We are listed on the Nasdaq First North growth market in Stockholm since 5 years now, and our current market cap is around 800 million Swedish crowns. We have a very strong shareholder base. Our main shareholder is Link, a Swedish life science investment company run by Bengt Jolander. We also have HPM, which is a well-known Swiss life science specialist investor. We have the Fourth National Pension Fund, and we have Handelsbanken funds. Together, they have about 40% of the company. But then we have a lot of smaller investors. In total, we have more than 5,000 shareholders in InDex. And to give you a little bit of background then on the lead asset Copytolimod, and Thomas will provide much more data on this. But it's a potential new medication for moderate to severe ulcerative colitis, the more difficult cases. And we met the primary endpoint in our phase 2b study, and Thomas will talk more to that. And we also have 4 previous completed clinical studies that support the efficacy and safety demonstrated in phase 2b, and Thomas will come back to that also. And based on the previous data, we are now advancing Copytolimod into phase 3. And what Copytolimod can offer is a competitive efficacy, well in line with the currently approved products and the late-stage pipeline from the industry, with a superior safety profile. And that's important because the current treatments are quite tough and associated with very severe side effects. This is a local treatment. We give it as an enema, as you see the bottle there on the right, every 3rd week. And it has a novel mechanism of action, which is also important, given that we would be alone with this mechanism of action. So all in all, we see Copytolimod has a very significant commercial potential, given the outstanding combination of efficacy and safety and the novel mechanism of action. And our estimate is that at the successful commercialization, the annual commercial sales could reach more than $1 billion. That's a bit of background. And today, this morning, we released our Q3 report. And I thought I would give some highlights. On the development side, we are now ready to initiate patient recruitment in the phase 3 study conclude with Copytolimod. We have activated sites, so they are now open to receive patients. So we look forward to welcome the first patient in the near term. In the competitive landscape in September, the FDA updated their safety warnings for the class of JAK inhibitors. So these are products. Well, there's one product that's been on the market for a while now, and based on a review of the safety data, the FDA actually added quite a few very serious side effects, including death, to the label of the JAK inhibitors, which really puts the focus on safety in this disease and for products in this disease. And that's really good news for us because Copytolimod, as Thomas will show, has an excellent safety profile. So there's a renewed or a strengthened focus on product safety in this area. We also could report a strengthened patent situation over the last quarter. We've had 2 new patents granted, one in the US and one in Canada. And this is the US patent is a good example of how we continuously file new patent applications to strengthen and extend the patent protection for Copytolimod. So we're building a patent thicket around this asset. And the US patent that was granted provides an exclusivity period until 2038, plus potentially 5 years of additional patent time. So it's quite far into the future now. Regarding the numbers, the operating loss amounted to $80 million for the first 9 months of the year. There is quite a lot of fluctuations in this because our costs are comes in milestones. We have if you look at this in a very short period, it's maybe not representative, but over 9 months it becomes more stable. And if you compare it to last year when it was 47 million, the increase in the loss is due to increased costs for phase 3 preparations. The cash and cash equivalents at the end of the period amounted to 463 million. That's 400 million more than last year this time. And that is explained by a rights issue we did earlier this year of more than 530 million Swedish crowns to fund the phase 3 development. So with this, I would like to hand over to Thomas Knittel, our Chief Medical Officer, who will walk you through our previous clinical data for Copytolimod in ulcerative colitis.
Thanks a lot, Peter.
Thank you, Peter, and also a warm welcome from my side. And I'm happy to introduce you into the clinical data we have generated over the last couple of years with Copytolimod. And so we have done a number of clinical studies with this compound, and the latest study we have done was the so-called CONDUCT study. And on this slide, you see the design of this study in which we tested Copytolimod in patients with moderate to severe active left-sided ulcerative colitis. For the ones which are not so familiar with this disease, it belongs to these inflammatory bowel diseases. There are 2 major forms. One is Crohn's disease, and the other type is ulcerative colitis, which is a chronic inflammation of your colon, so of your large intestine, and which starts at the very end of the colon and then can actually affect the whole colon, and then it's an extensive disease. The majority of the cases are left-sided, and this is where we test our drug. So we have studied Copytolimod in patients which have failed the standard therapy, which consists of 5-ASA and steroids, and also in more severe cases, immunomodulators, older immunomodulators, and so-called biologics. Here it's mainly antibodies directed against an inflammatory mediator called TNF, so anti-TNF antibodies, and also Bedo, which targets integrins in the gut. So patients which failed these more or less standard therapies were allowed to enter our conduct trial. And this conduct trial was done with the aim to identify the best dose and the best dose frequency to give Copytolimod to patients with ulcerative colitis. And on this chart or this diagram, you can see that we have tested Copytolimod in 3 different doses: 30 milligrams, 125 milligrams, and 250 milligrams, given 2 times as an enema at week 0 and week 3. And in one arm, we tested a higher frequency. So here we have given 4 times the enema, 125 milligrams, also adding up in total to 500 milligram. So with the last 2 arms, the 2 times 250 milligram arm, which is 500 milligram, plus the 4 times 125 milligram arm, we can really decide what is better. If we give the same dose, what is more important, to give a higher dose or give a lower dose with a higher dose frequency? Because this was completely unclear when we designed this trial. And all this was done in comparison to a group of patients which received placebo. This is shown in the lower part. These patients received 4 enemas with placebo, and placebo means water. Copytolimod, our drug, is dissolved in water and then given as an enema, and therefore the placebo is just water. So it was an exploratory study to identify the best dose and the best dosing regimen for a phase 3 trial later on. So these patients were treated, and then the effect of this treatment was analyzed at week 6 of the trial with our primary endpoint, which is called clinical remission. So patients went into the trial, we had a bloody diarrhea, we were really sick, we were ill, then we were treated, and then you look at week 6, how many of these patients got into remission. And remission means that these patients do not have any more blood in the stool, we have a dramatically reduced stool frequency, and we are getting better when you do endoscopy in these patients. And here now the result of the endpoint, our primary endpoint, is shown. As I said, the primary endpoint was defined as clinical remission with no rectal bleeding, no reduced or normalized stool frequency, and an endoscopic score of 0 or 1, which means endoscopic healing. So when you inspect the rectum, the sigma, the colon with an endoscope, then the inflammation is heavily reduced. And as I said, this was done in comparison to placebo, and in 6.8% of the placebo-treated patients, these patients get actually into remission, which is normal for the field. So these patients were kept under standard therapy. And also in other trials, this is a number which is absolutely common in the field, that a certain number of patients, when you put them into a clinical trial and they get really good treatment and good overview in this trial, they improve and get into remission. So our drug in the dose of 2 times 250 milligram, actually 21.4% of the patients got into clinical remission. So a lot more than with placebo. So 14.6% is the delta when we compare the effect of our drug to placebo, which does not sound a lot, but which is absolutely normal and in this field. So everything above 10%, also in case of other drugs, is regarded as a success in treatment. And when you look into other trials with other compounds, you find deltas of sometimes 8, 9%, sometimes 15%, sometimes 20%. So this is a number which is common in the field and indicates that your drug is effective. And you do, of course, statistical testing. And our study, now it goes a little bit in a statistical direction, was powered because it was a dose-finding study. It was powered within a one-sided test to have a p-value of less than 0.1, and once you're below 0.1, this is regarded in a one-sided test as statistical significant. And you see in this high dose group we achieved a value of 0.02, so highly statistical significant. And we even made it in a so-called two-sided test to get a p-value of less than 0.005. Here in our case it was 0.0495, so just below 0.05, which is the holy grail in statistics and clinical trials, because everything below 0.05 is regarded as statistical significant. So we met our primary endpoint with the high dose. We had a delta to placebo of 15%, which is pretty good in this field. This was reached with statistical significance. And this you see in the lower part of the slide, we also checked, of course, other endpoints, so-called secondary endpoints. And here in certain secondary endpoints, we also have statistical results and thereby supporting findings also in these secondary endpoints. So the drug is effective and it is pretty safe. And here you, in this overview, if you go through the numbers, you see that what we call adverse events, this is pretty evenly distributed between all dose groups, and this is important also compared to placebo. You see in placebo 21 of the 44 patients reported adverse events, 47%, and you see that in all the Copytolimod groups the percentage and also the absolute numbers are lower. If adverse events are more serious, for example, they require hospitalization, then this is called a serious adverse event. And this happened in 10 patients in this study. And you see again, it's found in all dose groups and also in the placebo group. And 8 of these 10 events, we should also mention, were caused by a worsening of the ulcerative colitis, which is logical. That when a patient comes into a study, of course, his ulcerative colitis can get worse. But in this system, it is called and it is regarded as a serious adverse event. So this serious adverse events, you should keep this in mind when you have a look on these numbers. We have seen one death in the study. This happened in the placebo group. This was a patient which underwent colectomy. So he, because his ulcerative colitis was so immense that the colon had to be removed, and then he has post-operative problems. But that happened in the placebo group. So you see the safety profile, we think it's pretty nice. And I think this also differentiates us from a lot of other compounds which are in development, because they have a lot more pronounced effect on adverse events. And I think this is also due to the fact that our compound is delivered as an enema, so it is more a gut or local therapy. We know that our compound is absorbed into the body, into the systemic circulation, only in very, very small quantities, and therefore I think this is one of the reasons why our drug has such a nice safety profile. The results have been published in a quite prestigious journal, Lancet Gastroenterology and Hepatology. So it's a subjournal of this super journal Lancet, which focuses on IBD, gastro and liver diseases. We actually also received a small editorial comment in this journal, in which 2 completely independent authors, which have nothing to do with InDex, nothing to do with the trial, have put our results into perspective, what it actually means for the field. And you see these highlights: gut selective therapy, because we have gut selective effect, and also a localized delivery of the compound. And these results were not only published in the Lancet, we of course presented them at several conferences, the UEGW, which is the European gastro meeting, and Raja Atreya, the main author of this paper and also the principal investigator of our study, presented the results. So I've shown you some now of the results of Copytolimod in the CONDUCT study with our 14.6% delta. This is on the very far right, with a statistical significant p-value. But this was not a one-time observation. We have done a number of clinical trials before in which Copytolimod was administered, sometimes in different concentrations, sometimes also the mode of administration was different. In CONDUCT, it was really an enema, how it will be later on used when the product comes to the market. In other trials, it was actually sprayed during endoscopy or administered with a catheter. So we are not 100% comparable, these trials, but the delta we see between the placebo effect and the Copytolimod effect is always in the same range. So it was a 14.6% delta in the CONDUCT study. In the COLLECT study, in which Copytolimod was sprayed during endoscopy, we had a 15% delta after 4 weeks when we looked at clinical remission again with a p-value. And also in the older studies, we have deltas at 9.5%, 13.6% in this other study. So this, of course, gives us confidence that that also the next study will result in a delta in these dimensions. So let me summarize. So in our latest dose-finding study, we met the primary endpoint. When we look on other efficacy markers with secondary endpoints, we also have supporting findings. With this 15% delta, we have clinical relevant efficacy. Everything above 10% is regarded as clinical relevant. And in contrast to a lot of other products, we have an excellent safety profile. We identified the dose and the dose frequency for further development, so 2 times 250 milligram given as an enema. And this was not only seen now in our CONDUCT study, this positive effect, but also in the previous, also placebo-controlled clinical trials. So based on the data of the CONDUCT study, we designed our phase 3 development program, which is called CONCLUDE. And as Peter said, the sites are now activated and ready to screen patients and put patients into the study, in which also the same group of patients will receive a dose of 2 times 250 milligram Copytolimod. We have one further arm with 2 times 500 milligram Copytolimod to get these patients into clinical remission. So this was a quick dive from my side into the clinical data we have. And now I hand over to either Peter or to you.
Maybe Klaus.
Yeah. Thanks a lot, Thomas. There's a question here from the audience. What type of adverse events is most commonly reported in the CONDUCT study?
In the CONDUCT, well, we do not see really any specific adverse event if you compare it to placebo. So we do not have something which occurs more common than in the placebo group. So there is not really a signal for a certain specific adverse event. It's all types, but always comparable to placebo.
Okay. Thanks a lot. Thomas. And you with your statistics here, which is very strong, it's interesting to see that if you take the risk reward and compare it to the way you dose it, so you have a 2 times or 4 times study, that you have a quite good effect on when you dose 2 times in this trial.
Exactly. I mean, this trial, one of the goals in this trial was to answer, if we have 500 milligrams on hand, what is better, to give it 2 times in a 250 milligram dose or 4 times in a 125 milligram dose. And I simply switch back, and
Yes.
The result was pretty clear that
Yeah.
it is better to give it 2 times instead of 4 times. I mean, this was a dose-finding study. What's also good news is that the highest dose had a clear effect. The lower doses had, as you see on these p-values and also on the deltas, not at least with these 43 patients we analyzed, we do not see an effect.
No.
So the high dose has a clear effect, lower doses no effect in this study, in this patient population with this number of patients. And of course, the result answered our questions. What is the best dose and dose frequency to go into phase 3?
Of course. And then just another question here. How many clinics did carry out the studies in the conduct studio? Because you mentioned something about that in the conclude studio, which is of course a much broader studio. But for how many clinics?
It was about 80 clinics all over Europe. So it was a study done in Western Europe, Central Europe, and Eastern Europe. And this is the number of centers you actually need to recruit these patients. So we had in total, I will go back, 213 patients which went into the study. And just to give you a feeling, to receive these, to get or to recruit these 213 patients into the study, you have to screen basically 400 patients.
Okay.
Because we have to fulfill the inclusion criteria to be very strict.
I know that both Peter, but also the company in general, mentioned that that it's mostly this disease is mostly in the Western countries, not European, Western countries, as we know of today. Can you say? I know I asked Peter this question before, but now with you, Thomas, maybe you can add something on. Is this lifestyle related? Because we know that some diseases like diabetes is very much lifestyle related. Can you say something about that? The current state of information you have as of today.
It is lifestyle related, and it is hygiene related. So in Europe, for example, we see a clear gradient between Northern Europe and Southern Europe. So ulcerative colitis is a lot more common in the northern parts of Europe compared to the southern parts in Europe. It's more or less unknown in Africa. So so the cleaner your environment, the higher is the risk that you get ulcerative colitis.
Wow.
And still the pathophysiology, so the reason why you develop ulcerative colitis are not 100% clear, 100% understood. It seems to be a mixture of your personal setup, of diets, of your surroundings. Maybe it is triggered by an infection sometime when you grow up. It's also discussed that certain bacteria in your gut might trigger this disease. So it has a number of factors which can trigger this disease. Even when I mean, I'm a gastroenterologist, so I have treated these patients. And the classical patient is because you develop it very early in your life. So you get it when you're 20, in your 20s, let's say. And so if you have a student and exams are coming up and we have stress, we often have a reactivation of either ulcerative colitis or of Crohn's. So even these psychological factors, stress, can activate this disease.
Interesting, very interesting. Peter, I know we will have a session sooner in respect of your next study. Well, there's another question coming in here, but maybe you could just very briefly elaborate on that, Peter.
Yeah, so we had our introductory event to InDex a couple of weeks ago, and this was now a deep dive in the previous clinical data, and then we are planning the next deep dive to be around the design of the phase 3 study and the setup of the phase 3 study.
Because it links very good to the next question and also my question in respect to this slide. If there's no serious side effects with the current dose level, why not increase the dose level to make the product even more effective? That's a very good question, I think.
But so the reason we went up to 250 as the maximum dose in the previous study was that at the time, that's what our preclinical toxicology studies allowed us to do. So that's what the regulators could allow us to go up to. Since then, we have done additional toxicological studies. So now we can actually go up to 500 milligrams. But above that, it starts to get a bit tricky because it's difficult to test it in tox studies or in animal studies because we are at such high concentrations now that you cannot fit so much more into a mouse or a rat. So
okay.
That's the reason. But even when we give very high amounts, we have not seen any negative effects of it. But in humans, it then becomes, your animal studies lets you translate to a maximum dose in humans, and that's somewhere around 500 milligrams now.
Yeah.
And actually, we test now as it is proposed, we test the higher dose. So we test now in the phase 3 study not only to the 250 milligram dose, but also the 500 milligram.
Yeah, I know that. Well, thanks a lot, Thomas and Peter. I don't think there's any more questions here from the audience. I don't have further questions either. I just look forward to host you, Peter, on our small mid-cap day in December, 8th and 9th of December, and also the next event where we make a deep dive into the conclude study. So with that, I would like to thank the participants, the audience, and I would like to thank both you, Thomas, and Peter as well. And well, that's all for now. Thanks a lot.
Thank you.
Thank you.
Welcome.